Hello everybody, I am Wolfgang Brill and I would like to present my proposal on Titanocene Conjugates as PET-Tracer and therapeutic agents. The concept of this proposal is using the same chemical entity for either PET imaging and therapeutic use. The chemical entity is composed of two parts: a ligand which is selective for diseased tissue, and a tracer structure that can be charged with either radioactive fluorine-18 or non-radioactive fluorine-19. In case of PET imaging, the radioactive fluorine-18 is being used, but the dose employed is below onset of biological activity. In case of the therapeutic application, larger doses are required, but non-radioactive fluorine-19 will be employed. The advantage of having an imaging tracer and a therapeutic agent with the same structure is evident. Their ADME and tox characteristics are the same. This means that the imaging experiment shows exactly the distribution of the therapeutic agent in the patient, which leads to better assessment of side effects and toxicity, and also facilitates the development. At the end, there is an improved safety for the patient. The chemical entity called for must bind to fluoride under mild conditions, which technically simplifies the imaging application where highly radioactive fluorine-18 is being employed. It must have sufficient stability to biological fluids. Degradation products would lead to a dissipation of label during the imaging experiments, leading to blurred images. In the therapeutic approach, degradation products may lead to additional toxic effects. The chemical entity must also interact most selectively with the diseased tissue, and it must become cleared out of the organism in order not to build up and cause chronic effects. One possible approach to meet this need is shown on this slide. It is a titanocene residue, which can be easily conjugated through its cyclopentadienyl groups with recognition elements for diseased tissue. These could be antibodies, carbohydrates, nucleic acids, or synthetic molecules. Titanocenes also display a high affinity to fluorine, which may actually lead to a simple conjugation with either radioactive or non-radioactive fluorine isotopes. How do titanocene species interact with cells? Titanocene dihalides without any alkyl substituents on their cyclopentadienyl rings hydrolyze quickly. Their cytotoxicity is caused by the shedding of titanium ions. When released in the vicinity of cells, titanium ions are believed to be taken up by a transferrin-based mechanism which places them in endosomes and allows their distribution throughout phosphate-rich structures inside the cell where they cause cytotoxicity. In turn, titanocene species with alkyl substituents on their cyclopentadienyl rings hydrolyze much slower. This allows passive uptake or uptake through other mechanisms. When titanium species are released distant from cells, titanium ions form aggregates with different biological activity. This slide shows the hydrolysis of titanocenes, which is crucial for their cytotoxicity. It begins by the substitution of the halides by water. The corresponding titanocene dihydroxides are rather acidic and shed cyclopentadiene. The resulting hydrotitanoic acid polymerizes to form titanium oxide, which eventually is not bioavailable. This is a summary slide on titanium uptake. First, there is passive uptake. titanium complexes of a certain log P value first may bind to albumin prior to diffusion through the cell membrane into the cell. This process is determined by Nernst distribution law. Second, there is uptake via various receptors. This process requires recognition elements being either the ligands of the titanium complex or the titanium atom itself. In the latter case, it must be exposable by dissociation of labile ligands. Third, there is uptake of titanium oxide nanoparticles, the ultimate solvolysis products of labile titanium complexes. This uptake is facilitated by liposaccharides or receptors. In macrophages and dendritic cells, this process may lead to the liberation of pro-inflammatory cytokines. Here we see the impact of passive uptake on the left and receptor-mediated uptake on the right. On the left, addition of serum proteins with unpolar clefts drive the distribution of unpolar titanocean Y. In fact, they extract titanocean Y from the largely more polar cellular interior. On the right, titanium silane, a good substrate for the transferrin receptor uptake system, pumps the titanium complex into the cell. This process requires energy, serum transferrin, and the transferrin receptor on the cell surface. Many titanocenes and other titanium complexes are substrates for the transferrin receptor uptake system. This usually serves as an iron shuttle. Thus, titanium and iron compete for the same receptor. In case of titanocenes, which may be hydrolytically labile, it is not known at which stage of hydrolysis they may be tolerated by the uptake system. For instance, whether both cyclopentadienyl residues or only one cyclopentadienyl residue is best tolerated. This slide shows the effect on a recognition element attached to titanocean dihalides on their cytotoxicity. Hodek et al. Published in their Organometallics paper 2014 various titanocean dichlorides or difluorides attached to carbohydrate residues. When those carbohydrate residues allowed interaction with D-glucose transporters, which are overexpressed in tumor cells, they were taken up more readily, boosting their cytotoxicity. The paper also shows that titanocean difluorides were more efficacious than titanocean dichlorides. Unfortunately, this paper did not provide any animal data. This slide shows the effect of the uptake of titanium citrates dissolved in serum on the two diagrams on the left, and the effective uptake of titanium dioxide nanoparticles as shown in the diagram on the right. Citrates are probably converted to transferrin complexes upon dissolution with serum, and then taken up. Already at 1 microgram per milliliter, this causes stress on macrophage-like cells, indicated by a gain of superoxide dismutase activity. Intriguingly, titanium dioxide nanoparticles exert the same effect in HFOB 1.19 cells. Higher concentrations lead to loss of superoxide dismutate activity. Thus, even the hydrolysis products of hydrolyzable titanocenes should be considered. What are the characteristics of titanocenes? The answer to this is extremely important, as it defines their potential added value over other drugs. Titanium-4 compounds do not oxidize themselves upon exposure to oxidants like peroxides or superoxides. However, they form stable complexes reversibly. This is an inherent feature of titanium, found in many compounds from titanium dioxide, titanates, and titanocenes. In fact, titanocenes bearing peroxide ligands have been shown to generate radical oxygen species called ROS. Another feature of titanium compounds is their ability to bind phosphates. This may provide an advantage over organic amine residues found generally in conventional drugs. The inherent features of the titanocenes should be considered when propagating their development as potential drugs. This slide shows how their potential as redox shuttle may be exploited. On the left of this slide, we see a titanocene dihydroxide, a possible intermediate of titanocene dihalide hydrolysis. After uptake, it may translocate into the mitochondria, as evidenced by atomic absorption spectroscopy or energy-filtered transmission electron microscopy. There, it may inhibit superoxide dismutase and related scavenging proteins for radical oxygen species, or ROS. This leads to an increase of peroxide being trapped by titanium. Due to their unpolar nature, the resulting titanocene peroxide compounds may travel to the nucleus where they may bind to DNA. Their ability to shuttle peroxide may enhance the oxidation of the chromatin, setting off DNA damage response, or DDR, eventually leading to apoptosis. Is there conflicting biological data on titanocenes? In cellular assays, their cytotoxic activity appears to be roughly directly proportional with the number of organic residues on their cyclopentadienyl rings. This seems to be correlated with their hydrolytic stability, being higher for titanocenes with more alkyl substituents. In animals, however, titanocenes with fewer alkyl substituents and lower hydrolytic stability had been shown to be the more active. How can that be? The problems generally lie in cellular assays that do not resemble the settings within an organism. The cellular assays should mimic the uptake systems, which include serum components, functional surface receptors, and downstream processes. Receptor-mediated uptake is strongly regulated. Cells being part of living tissue interact and behave differently than cells in a culture medium. In case of labile titanocenes, diverse hydrolysis products have different bioavailability and activity. The situation in circulating body fluids differs strongly from that in a 96-well plate. Finally, animal models should include a functional immune system. Another possibility is the immune response triggered by titanium dioxide nanoparticles. The latter have been shown to lead to the secretion of pro-inflammatory cytokines such as interleukin-1 from THP1 cells primed with liposaccharides. After removal of the nanoparticles by filtration, the interleukin-1 secretion was severely reduced. Unsubstituted titanocene dichloride decomposes quickly in serum. It can be either trapped by serum proteins or it aggregates to form polytitanates, leading eventually to titanium dioxide nanoparticles. On the left, we see how titanium dioxide nanoparticles could be removed by filtration. On the right, we can see the decrease of the titanium concentration originating from titanocene dichloride in human plasma. It is evident that most of the titanium-4 was rapidly confined to proteins or nanoparticles. This slide evidences the role of titanium oxide nanoparticles as stimulants for the immune system. When freshly prepared titanium oxide nanoparticles were subjected to murine macrophages stimulated with liposaccharide, phagocytic activity was induced. Also, titanium oxide was able to induce the production of cytokines and reactive nitrogen-oxygen species. The authors also claimed that titanium oxide did not induce DNA strand breaks in the priorly activated macrophages. This may serve as a support for the redox shuttle mechanism of titanocenes proposed earlier. Titanium dioxide nanoparticles inhibit superoxide dismutase but may not be able to interact as well with chromosomal DNA in the nucleus as titanocenes may do. Therefore, the ability to transfer radical oxygen species to DNA is not as efficient, and fewer lesions are observed. Considering the activity of titanium dioxide nanoparticles, which are possible hydrolysates of titanocene dichloride, may lead to another mechanism of action, found particularly in syngeneic mouse models. Some of the data was obtained with highly antigenic tumors. It is likely that the stimulation of the mouse immune system may have had a contribution on the overall tumor control. In turn, models using nude mice mirror the biological effect without contribution of the immune system. For the titanocene-mediated neutralization of cancer cells, titanocene difluorides should be used that are conjugated with recognition elements for tumor markers. This allows selective targeting of tumors, selective imaging of the diseased tissue, and providing a high concentration of titanium in the vicinity of the diseased tissue. The recognition element should also be highly antigenic to co-stimulate the immune response together with the hydrolysis products of the titanocene, the titanium oxide nanoparticles. The recognition element should allow either internalization of the titanium into the cell, either as a whole titanocene or by detachment of the titanocene residue. Only when the titanocene or the titanium is entering the cells, it can display its cytotoxicity. The latter is useful to assist the immune response by superseding the inhibition of apoptosis present in many tumors. Now we come to the chemical axis of titanocene conjugates. Their key intermediate is the stable titanocene precursor seen in the box on the right. It can be obtained upon treatment of various cyclopentadienyl anions with titanium tetrachloride, giving a cyclopentadienyl trichlorotitanium. The latter can be trapped by another cyclopentadienyl anion to give the titanocene dichloride, which can then be trapped with benzene dithiol to give the precursor. The benzene dithiol ligand allows aqueous workup and chromatography on silica. It also provides high fluoridation rates. The latter are being particularly useful when applied in PET imaging. The nature of the alkyl groups on the cyclopentadienyl rings allows fine-tuning of the hydrolysis and fluoridation rates. This slide shows the flexibility and convenience of the titanocene precursor, whose synthesis we have discussed in the previous slide. On the left of that slide, we see such a precursor molecule bearing a protective carboxylate function. This carboxylate can be deprotected and the precursor then coupled using carbodiamide chemistry or a Staudinger coupling reaction as described by Bartosi to a recognition element. The carboxylate function may also be modified to access other coupling chemistries like metal-free Plick chemistry or NHS ester couplings. The conversion to the final product is obtained by displacing the benzene dithiol ligand on the titanocene by fluoride. This can be performed prior or after the conjugation with the recognition element. Substitution of the benzene dithiol residue by radioactive fluoride 18 will provide the PET imaging tracer. Displacement of the benzene dithiol residue by non-radioactive fluoride will yield the chemotherapeutic agent. The simplicity and flexibility of the titanocene synthesis allows rapid optimization of the titanocene structure towards hydrolysis rate and fluoridation rate. The latter two rates depend strongly on the number of alkyl substituents on the cyclopentadienyl rings. The slide also shows the effect of benzene dithiol as optimal leaving group. The flexible synthesis of titanocene conjugates may be used to synthesize titanocenes with diverse ligands by an Ugi reaction. A benzene dithiol group with an nonpolar or fluorous functionality may facilitate a high-throughput purification. Prior to the synthesis, the expected structures may be docked onto targets in silico. The most promising candidates may then be synthesized and tested in a biological assay with a functional uptake system. On the bottom of this slide, we see a rough idea for a pharmacophore model for ROS scavengers on the left and RNA structures or ATP binding sites in kinases on the right. Here are some considerations on the use of titanocene derivatives as PET tracer. First of all, the half-life of the F18 isotope is 109.77 minutes. It decays by positron emission 97% of the time and electron capture by 3% of the time. Fluoridation of titanocene and workup must therefore be performed in much less than 330 minutes or 3 half-lives. A conjugation with fluorine of less than an hour is of course more desirable, as the label would be stronger. The benzene dithiol ligand may be very suitable to achieve this goal, as it can rapidly be displaced by fluoride. After injection, hydrolysis of the titanocene fluoride will occur. It should be slow enough in order not to dissipate too much of the label. A recognition element for binding to deceased tissue prior to hydrolysis must be found. Suitable antigenic structures are in phase 3 in the industry. Here we see the potential effect of the proposed titanocene conjugates on PET imaging. On the left we see traditional PET performed with FDG. FDG evidences only tissue with high metabolic rate. Of course, tumors have high metabolic rates, but also other tissue will also be evidenced. On the right, however, we would see an effect of a titanocene-based tracer interacting specifically with a tumor marker. Only tissue bearing this specific tumor marker would be evidenced. With this in hand, we may consider a titanium-mediated therapy. It starts off with smart PET. The titanocene conjugate will interact selectively with the tumor through the interaction of its recognition element with a tumor marker. In this case, very small amounts of the titanocene conjugate, however charged with radioactive fluorine-18, will be used. For the therapeutic purpose, the same compound will be used at larger doses, however charged with non-radioactive fluorine-19. En route to the tumor tissue, where it will be concentrated due to its high affinity to the tumor marker, it will set off the immune response. Once reached the tumor tissue, it will display its cytotoxicity. This titanium-based cytotoxicity will assist the immune system in combating the tumor. In conclusion, titanocene conjugates are likely to act as PET tracers and anti-cancer drugs. As PET tracers, they are likely to be fluorinated under milder conditions than existing imaging tracers. This facilitates labeling and increases the safety for the personnel that performs the labeling. Titanocene conjugates act by cytotoxicity and stimulation of the immune system, the latter by their recognition element and their degradation products, the titanium oxide nanoparticles. Titanocene dichloride had been used in phase 1 clinical trial. It failed due to lack of conjugation to a recognition element for tumor marker to provide an antigenic complex directing the immune response. Lack of this conjugation would also lead to a greater hydrolytic lability that prevents selective targeting to diseased cells to display a selective cytotoxic effect. Titanocene syntons for fine-tuning the reactivity are described. Potential antigenic recognition elements are in phase 3 clinical trials throughout the industry. Titanocene conjugates would enrich many portfolios in industry, particularly if entities are available that could serve as recognition elements. Their development is foreseeable owing at the vast amount of data available in the literature. It would provide an improved imaging and therapeutic approach, giving at least a better prognosis and a better monitoring of the therapeutic outcome, serving the patient. Do not hesitate to contact me on this topic, and thank you very much for spending your time. Goodbye.